Welcome to BioIT World's Trends from the Trenches podcast, your insider's look at the science, technology, and executive trends driving the life sciences. I'm Allison Proffitt, editor of BioIT World. This episode is hosted by Stan Gloss, founder of BioTeam, a life sciences IT consulting company at the intersection of science, data, and technology. Joining Stan today is Tom Miller, co-founder and CEO of Iambic Therapeutics. Together, they discuss Miller's journey from academia at CalTech to CEO of his own startup and what inspired him to turn his research into an entrepreneurial venture. Let's listen in.
Well, good afternoon, Tom. I'm so honored to have you here, telling a little bit about the story of your company. And boy, we haven't had enough startup stories yet. So I'm really excited to really get into how the journey that you've had from your academic work all the way into B-round funding as a startup. So perhaps we can start by getting a little bit about your background and then take us through part of the journey of your research and maybe even talk about the moment that you said, gee, this could be commercial. Because I'm sure a lot of scientists listen to this and want to hear those same questions.
Absolutely, Stan. Thank you so much for the opportunity to join this podcast and to tell you about Iambic. And I'm happy to kick it off with some description of my background. I am Tom Miller. I am one of the co-founders and the CEO of Iambic. And I arrived at this role after a career in academics. I started at the interface of chemistry and mathematics in terms of my training. I was a professor at CalTech starting in 2008 and really had the chance to build a research program there with many exciting research directions that focused on developing computational and AI or data-driven algorithms to better solve a range of chemistry and biological challenges with many different research focuses along the way. And what we really increasingly saw during my time at CalTech was the way in which these tools were not only exciting scientific innovations in and of themselves, but they had an increasingly translational capability. And to come back to your question about what was the moment or what was it that really triggered the curiosity to turn this into a startup or in terms of an entrepreneurial venture, I think it was, first of all, that hunger to see the translation of the science from the discovery stage to genuine human impact, as well as the recognition that these tools really did appear to have the ability to make a difference in the way that we were searching chemical space, in the way that we were identifying molecules with promising properties. And I think that connection between that desire and that opportunity was a fertile ground for launching a startup.
So what I didn't hear in your background is an MBA from a major school or previous startup experience. And I hear the story of an amazing academic researcher who went from the bench to the C-suite and in a direct path almost. Is that true?
I think in a nutshell, that pretty much is true. I have my fair share of degrees, but they are all on the scientific side. I did my PhD at Oxford and I did a postdoc at Berkeley. I did undergraduate at Texas A&M, and those are all focused on chemistry or mathematics or the conjunction of the two. I did not have prior executive experience. I had not founded multiple previous companies. This was a new adventure for me as an individual. And I must say that CalTech as an institution was very supportive of giving me the space and opportunity to explore that. I had long been a tenured professor at CalTech, and I hadn't taken a sabbatical yet. So they were very kind to give me 2 years of sabbatical so that I could, launched the company along with my co-founder, Fred Manby, to see whether or not the ideas had legs, to see whether or not my interest in an operational role within the company was of interest to me and was effective and good for the company, and to really kind of test drive it before making that commitment that I eventually did.
Right. So at the center of really understanding a company, for me, is understanding the why. Why did you do this? And the big picture of why it matters to researchers and the people who you plan to is the people who you're going to solve a problem for. So what was that problem, and what's the why, your company why?
Yeah. I mean, the why is, why can't we deliver molecules to patients with urgent medical need faster and more successfully? That's the why. And there are many things that make that an extremely challenging process. And one of them is the challenge of being able to identify chemical matter, new molecular therapeutics that can have the array of necessary properties and that can have the quality as development candidates to address those needs. And that involves an extremely vast search of chemical space and an extremely vast search of biological target profiles. And AI, in combination with high throughput experimentation, offers an extremely powerful way to try to navigate that bottleneck in delivering better molecules to patients faster.
Wow. That story reminds me of the story that Chris Gibson from Recursion Pharmaceutical, when he was on my podcast before, talking about this building up of his phenomics platform and doing millions of biological experiments to perturb cells to see what happens, and then grab imaging and building a huge training data set that now you can look for signals in that. So you did a great job of explaining the why of your company. So the next question easily follows, how are you doing it? And
that's exactly right. And of course, Recursion's an inspiring and relevant, marker on the landscape for how to contextualize a company like Iambic. I think the how comes down to three aspects of differentiation for us as a company. And one of those is the key AI algorithms that we develop and that drive our design process. The other aspect of how we do it is to tightly integrate those AI designs with high throughput experimentation, automated chemistry, and biology that allows us to go from new molecular designs to new data on the scale of thousands of molecules every week. So new closed loop cycles on the 1 to 2 week time scale, so that we can ask and answer questions quickly. We can drive iterations through chemical space. We can optimize target product profiles, and we can arrive ultimately at better development candidates. And that third key aspect of differentiation is showing that it really matters, that it's showing that we can compress the timescales from program launch to IND submission, and to do that repeatedly across challenging targets, across different mechanisms of action, and across different protein target classes. And we see that with our lead programs. Our first program went from program launch to IND submission in 24 months. And that's faster than the industry average, which is closer to 5 or 6 years. And we're seeing that that's a repeated process with our second and third programs on the heels of that first program, and following at the same pace.
That's great. That really covers my, how. So now I want to take you down the what's the result? What do can you give us specific? Please don't share any confidential information, but specific. Can you give us a little specific about the outcomes? If you happen to own. So tell me a little of that story.
Yeah. So let me break that up into kind of two aspects of the specifics. I can talk about specifics around the AI technologies, and I can also talk about specifics around the actual drugs that have been produced on the basis of this platform.
Yeah, so
And
that'd be great. Do that.
Yeah, I'll do it in that order. In terms of our AI technologies, I think that the mention of recursion is a relevant one, because there are companies that will generate vast amounts of data, and that will use that data-driven approach to identify better target profiles, or to try to identify better molecules for development candidates.
Actually,
there's also
I want to just quickly jump in on that one. If you could do a Google search on the SEC S1 filing for Recursion Pharmaceutical, and just read the business section of their S1 submission, it's a beautiful story of exactly, Tom, what you're describing. And they're streamlining the discovery process by actually, instead of having a discovery funnel, which has been traditional over drug development for many, many years, they're approaching it as a T-shaped approach, which is they have lots of data up here, and they look for signals in their data, and then they come straight down into what they're drilling down directly into their signal and trying to better understand it. So, it's when you were describing it to me, it sounded very similar, like I heard that story before.
Yeah, so I think that's one of the reasons it's really important to make that distinction. I'm basically setting up a dichotomy. There are companies like Recursion that are data-driven. There are companies like Accenture that you have also interviewed that are also very data-driven. They basically will gather data, they'll build AI algorithms on the basis of that large amount of data, and they'll use that as a way to design profiles for molecules. On the other side of the coin, there are companies that you'll also be very familiar with, like Schrödinger and like Relay. Those are also computationally driven companies, but they are not data-driven. What they are is physics-driven. They will use molecular simulations. They'll use detailed modeling of the atoms and the molecules computationally to try to design molecular profiles. And this is the distinction that I want to make. Those are not disjoint, they should not be disjoint strategies, even though that is often the way in which they are implemented at present. It's not using physics modeling or using a purely abstracted data approach that is really the optimal strategy. What Iambic has done from the very earliest days is recognizing that you can get the best of those two worlds by using physics modeling or physics insights to construct more data efficient AI algorithms. It's not just a matter of relying on big data and hoping you have enough coverage of that chemical space for that data to be useful to you. And it's not just a matter of not using data at all and to simply use a pure physics modeling approach. By combining physics and data, we can get vastly improved accuracy in the prediction models with more efficiency in terms of the number of data points that are needed. And that really matters. That matters because of the fact that we can more effectively drive more deeply into chemical space. Chemical space is extraordinarily vast. You will never have coverage of that with any existing data set. So unless you can see more deeply into that design space with AI algorithms, you are never going to be able to fully search to the extent that will lead to the best possible molecules. So we really take that approach, the best of both worlds, by unifying data and physics in a singular approach at Iambic.
Yeah. So when I think of going back to I'm sorry we keep going back to recursion, but they've kind of flipped the hypothesis-driven research model. So data-driven companies, initially with traditional scientific method research, we formulate a hypotheses, and then we develop data to accept it or reject our hypotheses. In a lot of companies that are data-driven, they're building up a large amount of data first, then they're finding these signals, and from the signals they get their hypotheses. It's a little bit flipped from the traditional hypothesis-driven research. How does that model apply to your company?
We absolutely can pursue hypothesis-driven research, and we can absolutely incorporate maximum flexibility in that search based on new signals that emerge. And I should take a concrete example here. Our two lead programs and our small molecule TKIs. The first is a highly brain penetrant and highly selective HER2 inhibitor, and the second is a highly selective CDK2, CDK4 inhibitor. Those ultimate target product profiles that we arrived at were not the ones that were anticipated when we started that program. We had conviction around those targets, but the actual final molecular profile that optimized therapeutic window in vivo was not clearly known at the start of the project. It was only by being able, through rapid design make test cycles, to be able to make the compounds that tested different biological hypotheses that we were able to refine that target product profile and ultimately to converge at what are extremely exciting and differentiated molecules and target product profiles. So I think that incorporates both the hypothesis side of the question as well as that responsiveness to new signals through the ability to execute very fast design make test cycles and to efficiently peer deeply into chemical space with the use of our AI tools.
That's so perfect. Thank you. So you have the best of both worlds.
There's many wonderful companies out there, but that is really our strategy, to try to incorporate the best of those both worlds and to build a platform and approach that really can do that. And I think that what really matters at the end of the day, of course, is not the philosophy of the strategy or the philosophy of the platform. What really matters at the end of the day is the resulting chemical the development candidates that we're able to deliver to clinic. And it's worth noting that our brain penetrant HER2 program with activity not only on wild type, but also across all the disease-driving mutations of HER2 was executed from program launch to IND submission at a pace of 24 months. And our CDK2/4 program is operating at the same pace as our additional programs across different protein classes. So having highly differentiated molecules and being able to repeatedly execute on that at an exciting pace and arriving at those quality development candidates, I think is really the most important validation for us as a company at the end of the day.
So with all of that activity, I want to return back to your startup journey. Tell us a little bit about the transition from angel round investment, and you're now into your B round investment. And what's that been like?
Oh, it's been exciting. It's not my original analogy, but I do like it. This notion that as a company goes from a seed round to a Series A to a Series B, it feels a lot like going through different kind of grades in school, or kind of different kind of levels of advancement in terms of going from middle school to high school to college, and in the sense that that these are big steps and these are exciting steps, but you also know that they are the first steps in ultimately what is a long journey of education and maturation as a company. So I've really enjoyed that process. Maybe more specifically, our seed round was it was led by Nexus Ventures in the Bay Area. They had a strong connection with CalTech. and also joined by another VC, Free Flow, and they really supported our vision before we had identified even drug discovery as the key focus area for the technology. At that point, we had a very general platform for designing molecules using physics-informed AI. We saw many potential ways to kind of point that flashlight in terms of opportunity space. And we initially focused on, deploying actually the software itself as to different partners as the product. Within that first year,
go ahead.
Oh yeah. Just within that first year, what we were able to do is to recognize that not only does this appear to work, but it has a particular sweet spot in the space of drug discovery. So that triggered the logic for our Series A, which was co-led by Catalio and Co2 and joined by Orbimed and Sequoia, and allowed us to really convert that software basis into a closed loop drug discovery focused company that would deliver our own fully discovered molecule pipeline.
So back to the connection with Recursion. As I was visiting there, and I noticed they had made significant investments in their phenomics platform by purchasing a large amount of NVIDIA product. And I understand that there's an NVIDIA connection, again, with your company. Maybe you can kind of share a little bit about what you're doing with NVIDIA?
Oh, happy to. Yeah, NVIDIA has been a great partner and, to us as a company, and also I would say to healthcare and drug discovery as a field. We have strong and long-term ties actually with NVIDIA. One of my original academic collaborators is Anima Anand-Kumar, who is both a director at NVIDIA as well as on the CalTech faculty, and we're working together for many years. And with NVIDIA scientists over the past 4 years now, we've published numerous papers together. So it's been a very, tight actual intellectual collaboration that we've benefited from and as a company over that time. You're absolutely right that NVIDIA has joined our Series B as significant investors. And the Series B announcement was accompanied by a major collaboration between Iambic and NVIDIA for the upcoming years, really extending and building upon what we've already done together in terms of that collaboration foundation. And, that brings the opportunity to benefit from the engineering experience and expertise of the NVIDIA team. It brings the access to the necessary GPU compute that is essential for developing next generation large language models and really deploying those at scale. And it really puts us in a strong position for continuing to differentiate both on the algorithmic development and on the computational deployment of these AI models at a time where that's a key strategic consideration.
That's fantastic. My final question to you is your advice for academic researchers to kind of follow in your tracks, and what kind of advice can you give them about taking their discoveries and bringing them to market? Any insights on that?
I do have. I'm happy to share my experience, and I do have a couple of thoughts that are triggered by that question. I think that speaking to the academics out there, I would say that parts of creating a startup will feel very familiar to you. The part of starting something from scratch, the part of having to recruit and align a team around a vision, the part about having to make sure that you set priorities correctly, and that you are able to really secure the funding to continue on that vision. That's actually surprisingly aligned with the day-to-day of a university professor at a major research major research institution. I think that I would also say that the thing that's very different, of course, is that this is all without tenure. So it feels like the pre-tenure days. And some people really like that. And actually, I really like that. This idea that every month matters, every year matters. You can't think in terms of, just lackadaisical timescales. There's an urgency, and there is a progress that can be achieved at a very fast pace with that urgency. And I've absolutely loved that part of creating a startup and being part of this amazing team.
Well, listen, Tom, thank you so much. This was really fantastic, and the story is great. All the connections that you've shared and the amazing results that you've gotten, and I hope my audience can really learn quite a bit from you today. So thanks again, and we'll talk soon.
I've absolutely enjoyed it, Stan. Thank you so much.
Thank you for listening to BioIT World's Trends from the Trenches podcast.